Welcome students. In this video we will discuss about the different types of endosperm and how these different types of endosperm are developed. On the first and the subsequent division of the primary endosperm nucleus, endosperm are three types. Then what is this primary endosperm nucleus? In the central portion of the embryo sac, there is present a central cell and which contains the two nuclei or the two secondary nuclei, and these two nuclei are called as the polar nuclei. And with this central cell, the small male gamete unite and this will form a triploid structure, and the nucleus of this triploid cell is known as the primary endosperm nucleus. Okay, so this is the male gamete and this is the central cell which contains the two polar nuclei or the secondary nuclei, and after fertilization this will form a triploid structure and whose nuclei is known as the primary endosperm nucleus. Now, how this primary endosperm nucleus undergoes the further modification, this is very important, and this will causes the development of different type of endosperm. Actually, there are presents three types of endosperm in case of angiosperm. First is nuclear endosperm, second type is cellular endosperm, and the third type is Helobial endosperm. Now first, the nuclear endosperm, how this nuclear endosperm is developed. Here you can see this is a cell and which contain the triploid nuclei, and this triploid nuclei is the primary endosperm nucleus. And this primary endosperm nucleus undergoes the free nuclear division many times. Then this nucleus undergo many times of free nuclear division and this will form the many nuclei. Here you can see there is presence many nuclei. After this free nuclear division, a central vacuole is formed in this structure and this pushes all nuclei towards the periphery of the cell. So this all nuclei arranged towards the periphery of the cell, and this is the central vacuole. And in the next stage the wall formation is occurred. Okay, and this wall developed just outside of this nuclei, and this will causes the development of a multicellular hard structure, that is the endosperm. Okay, so here first nucleus is divided many times, and for this reason this is known as nuclear endosperm, and cytokinesis occurred later to the karyokinesis. And generally we see that after karyokinesis the cytokinesis occurred, but in these cases karyokinesis means the nuclear division, but in these cases first karyokinesis occurs many times and later the cytokinesis can be seen. Okay, so this is the procedure of development of nuclear endosperm, and this nuclear endosperm is the most common type of endosperm, and this is present in 161 angiosperm family. And this type of endosperm can be seen in areca nut plant, wheat plant, sunflower plant, and the coconut plant. It's the process of development of nuclear endosperm, and these are the examples where we can see the nuclear endosperm. And in the next type of endosperm, this is the cellular endosperm. Individual cell will be formed after karyokinesis, cytokinesis occurred. Okay, next again karyokinesis occurred, and then again cytokinesis occurred, and thus by this way many cells is formed, and this cell simultaneously produces many number of cells, and this will causes the development of cellular endosperm. So this is the primary endosperm nucleus. And first the nuclear division, this nuclear division followed by wall formation. So this primary endosperm nucleus undergoes the karyokinesis, and this will causes the development of the two nucleus, and then the wall is formed around this nucleus. Okay, that means from one cell the two cell will be produced. Okay, and later many times this undergoes mitosis cell division, or the karyokinesis and the cytokinesis occurs simultaneously. So cytokinesis occurs after each of the nuclear division, and this will causes the development of a multicellular triploid structure. This is known as the cellular endosperm. So in case of cellular endosperm, no flea in nuclear stage can be seen, and this type of cellular endosperm can be found in balsam plant, petunia, adoxa, and the datura. Okay, the next type, that is the third type of endosperm, is the Helobial type. This Helobial type of endosperm is very important for your exam purpose. Okay, so this Helobial type of endosperm can be seen in monocotyledons of order Helobials. Okay, then how this Helobial type of endosperm is developed? So this is the primary endosperm nucleus, and this primary endosperm nucleus undergoes the karyokinesis, and this will form the two chamber. One is large chamber, this is known as the large micropylar cell. So in the upper side you can see a large cell, so this cell is known as large micropylar cell, and in the lower region the small chalazal cell is formed. And After formation of these two unequal type of cell, the free nuclear division occurs in each chamber. Okay, so free nuclear division can be occurred in micropylar chamber, and also the free nuclear division can be seen in small cellular chamber, and this will causes the development or formation of many nuclei, but these nuclei are without the wall. Okay, so free nuclear division in both cell, and this will form the many nuclei, but this nuclei is devoid of wall. Later, wall develop in between nuclei of the micropylar chamber. So upper side is the micropylar chamber. Generally, this micropylar cell are formed towards the micropylar end. Okay, so in this micropylar chamber, the wall formation is can be seen, but in the cellular chamber, no wall is formed. Okay, so around the nuclei, no wall is formed in the cellular chamber, but wall formation can be seen in micropylar chamber, and this will causes the development of an intermediate type of endosperm. Okay, and this is seen in case of monocotyledons, and these are the procedure of development of three type of endosperm in case of angiosperm. Remember, the endosperm is very nutritive, and this provide nutrition to the embryo, and at first this endosperm is stored in the seed, and later for the further development, the development of the cotyledons or the first leaf of the plant is can be seen, and the endosperm is stored in these cotyledons. Okay, and usually endosperm contains the protein, starch and the oils, and this will provide the nourishment to the developing embryo or to the developing plant. Usually endosperm contains the protein, starch and the oils, and this will provide the nourishment to the developing embryo or to the developing plant.